we are very pleased to have three experienced speakers with lots of experience in trial designs and in master protocols. I'm also very pleased to have two speakers from the United Kingdom here today. Welcome. There's lots of experience with master protocols across the ocean in the United Kingdom, so I'm very happy that we can learn from their experience in the Netherlands today in this webinar. We will start soon with a talk by Matthew Sydes. He's a professor from clinical trials methodology in London, and he will talk about we will discuss platform protocols from a practical perspective. Thereafter, we will continue with Peter van der Ven from Senior Biostatistician Clinical Trials at the UMC Utrecht, and he will discuss master protocols from a statistical perspective. So how to analyze these trials, what are the challenges, and how to solve them. And we will end this webinar with a talk from a clinical perspective by Professor Andrew Biankin, who is not here at this moment, but he will tune in at 4:10, and then he will discuss the pragmatic aspects of precision oncology and drug development. And when we have time left at the end, there's still some time for discussion. So before we start, if you have a question, use the chat, and after each talk, we will try to answer as much questions as possible in the time left. Otherwise, we will move them to the end of the webinar. And if there are still some questions left at the end, please send them to us and we will try to get back to you in due time. I'll ask all of you to mute yourself, and a final note is that the presentations will be recorded, but the discussion sections won't. So now I'll stop sharing, and if there are no further questions at this time, I will ask Matthew to share his slides and welcome us in the magical world of platform protocols. Floor is yours, Matthew. Thank you.
Thanks, Rob. Can that be seen okay? Are the slides up okay?
Yes, the slides, we can see the slides, yes.
Brilliant. Okay, that's really good. It's just, that's working. Yeah, so thank you very much for having me here today. As Rob says, Matt Sydes, been around platform protocols for a number of years, and we're always very keen to share our lessons for what we think are sort of our very efficient designs. There's a number of the papers that are linked in here. There's a little QR code here or a shortcut to that list of papers. I'll put it back up again on the final slide. So if you want to reach out and grab that at the time, you'll find that it's all listed there, but I just wanted to flag that with you now. So I'm going to talk very much from a practical perspective about the implementation of MAMS, multi-arm, multi-stage platform protocols. And let's see if my mouse is going to play nicely. It is okay. So platform protocols, still here. You know, the master protocols, as Rob was saying in the introduction, is quite used. Living protocols are being used for a while as a term. Effectively, you know, what we're trying to do is use a clinical trial protocol to address many research questions within one administrative trial structure. And there's a number of good reasons why we might want to do that. One of the key things I think the platform element is the adding on of new important questions later on into a trial, rather than setting up a competing trial, rather than waiting to ask a question, rather than having somebody else set up a competing trial, then trying to incorporate that question in a sensible way as possible, or only in a sensible way, into the trial protocol is really important. There are plenty of ways in which that can be done not very well, and actually, you know, we're keen to avoid that. And even when we do it well, the practicalities can be a little difficult. So just want to run through some of the lessons. And, particularly in the context of oncology, you'll be thinking about stratified medicine, biomarker-directed therapies in the same disease. Yeah, I think that's really important for us to be bearing in mind here. So I'm going to start by a sort of motivating example for us is the Stampede trial. It's been running in the UK for about 20 years. There's Max Palmer, who's the director of the unit I work in, who's the originator of this approach. Nick James, who's been the chief investigator and managed to find a very appropriate science to stand next to when he's skiing. This is a trial for men with prostate cancer for which there were a number of potential treatments that could be looked at, but no progress had been made for many years. So the motivation was, why will we pick one of these treatments when there's a test, when there's a real need to move forward as quickly as possible for this particular patient group? So I say it opened as a multi-stage platform trial. You could see the multi-arm element when it started with 6 arms. If this is a framework of time across the bottom, the arms are listed down the on the left and right here. Orange represents future recruitment, green represents past recruitment. So orange here is in October 2005. We're planning to recruit for about 7 years, get answers after a further 2 or 3 years of follow-up. You can see this here. So this is very much a multi-arm trial. You see the multi-stage element, really, you see it kick in practice when at the second interim look, the data monitoring committee said, actually, 2 of your arms are not looking interesting enough. The MAMS approach says it's not just stopping for harm or stopping for superb effect. It's actually saying if something is interesting but not interesting enough, you still don't pursue it, particularly if you've got other things that you can be looking at. So you can see that 2 of the arms D and E here, stop recruitment early in the other arms, and the primary analysis for these gets pushed out back in time behind the other ones. So you see the multi-stage element, and you see the platform element really kicking in later on that same year when the first added arm came in, a test of another drug that was looking like it might be very interesting for this group of men with prostate cancer. Rather than have a competing trial, which was a possibility in the UK, better try to incorporate it. And you'll see that the key change here is that the control arm got lengthened here. The control arm got lengthened because we're only going to be looking at contemporaneously randomized patients or concurrently randomized patients with this approach. So this was November 2011. And actually, by just before the pandemic here, you can see that the trial then gone on to add another 5 or a further 4 comparisons in questions that were worthy of asking. And it was more efficient to put them into this protocol than to put them into a separate protocol. And why do we think this is a good idea? Well, we know every trial takes a long time to start up to recruitment. Every dot here is a patient that joined the trial. Same sort of structure here, but it's a dot. You can see the trial started slowly, partly because every trial starts slowly, partly because we deliberately limited the number of centres, partly because, as Max said when we were designing the study, it's a slightly mad idea, right? So of course it's always going to be a challenge to recruit to at first. But you see, I think you see the density pick up as recruitment comes on board. You see the density of the dots change when we stop recruiting to these 2 arms D and F. You can see how the density, the rate of patients should stay the same. So the dots just become a bit more dense on the others. You're effectively upping recruitment to those. If anything, stopping recruitment to these 2 boosted the credibility of the study, and recruitment was increased at that point. You see, when it drops down to 2 or 3 arms for a while, there's a lot of strong density of the dots here. And you also see, as we all see in our trials, the impact of the pandemic for a while here, where there's no patients, right, coming in. So, keeping that momentum rather than having to start again and again for trial is really important. So let's talk about site initiation. Along the bottom here, you've got the time to the first new patient being randomized from a site since a comparison was added. And along the y-axis, you've got the number of sites randomizing at least 1 patient. So for the original trial, for the original comparisons, when we opened it, it took us 60 months, it took us 5 years to get 80 centres to randomise at least 1 patient into the study. Right? So it takes a long time to get people up and running. When we added in the aberration comparison in November 2011, is this going to work for us? What you could see is we had 80 centres randomise at least 1 patient for which they could have been allocated to that comparison within 4 months. You hit the ground running to that comparison, so you don't have that startup time. And that's the same with each of the comparisons that's subsequently been added. Even this comparison, this yellow one here marked as TE2 comparison, it's slower than these other ones. It's slower because to be eligible for that comparison, the criteria are a bit more strict as to how much treatment you could have before you're able to join the study. So it was much more restrictive, and yet it was still, look, so much quicker to get sites on board than the original comparisons. So even when it was slow, it was much faster than setting up a standalone trial there. And we know that these trials can present practice-changing results, and I'm going to run through 3 of them very briefly because it's relevant for something we'll come back to later on. So this is just showing you how we've broken this up for analysis. This is standard of care, SOC, against adding docetaxel and prednisolone. So standard of care or standard of care with docetaxel and a supportive drug. And all we're talking about in this comparison here is the yellow people. Just the people in yellow, those patients who in the original comparison that made it through their 3 interim looks and completed recruitment. So we've got about 1,800 patients in this comparison. 1776, I think it was. It's a 2-to-1 allocation ratio. Recruitment was over 8 years. It was a 2-to-1 allocation ratio because we have 5 research arms, we loaded a few more patients into the control arm. And you can see that overall we had a hazard ratio of 0.78, 22% relative improvement in survival, which is roughly what we were targeting, and that was enough to start changing practice. We also showed very positive results in the first added comparison of abiraterone, just the yellow patients. I said it's just the contemporaneous patients here, and I tell you these recruit fast. We've got 1,917 patients here, 1,917 patients. We said we recruit between 1,500 and 1,800, and we'd recruit to a maximum of 1,500 or for 3 years, whichever came sooner. But we agreed with the company we could go to 1,800 patients, and we still ended up over recruiting because of a ridiculous final month to the study and finishing after 2.25 years. So that sense of momentum was much quicker than we anticipated. So again, positive results, hazard ratio of 0.63 here. And the company actually did go off and do their own study. They just didn't run it in the UK, and they showed pretty much the same hazard ratio. They showed 0.62 in a high-risk subset of these patients. And again, radiotherapy to the prostate. This was just for men with metastatic disease, so it's not all of the patients. This was effectively phenotypically stratifying the patients. So stratified by whether they have metastases, you could join this comparison. You can see it's just the patients in yellow, just contemporaneously randomized patients. 2,061 patients here in this comparison, but there was no improvement overall with in overall survival, 0.92. This one did have a pre-planned subgroup analyses based on a Dutch study that looked at a smaller Dutch study that reported a couple of years before that thought the radiotherapy might only work in a subset of patients. We showed exactly the same results as that, but in a slightly larger population. So together, again, they've changed practice in low burden metastatic disease. So we can get practice changing results here. And this trial, as we summarize it here, is effectively running 10 major phase 3 randomized trials against a standard control over 20 years, which I think is quite remarkable for any single protocol to do. So, but there's always lessons to learn from this, and we can learn from Stampede, we can learn from other trials. Here's Nuru Noor, he's been doing a PhD with us. He's a gastroenterologist at the unit. He did a review of the number of multi-arm, multi-stage trials. So not necessarily the platform, but multi-arm, multi-stage. And he sort of reviewed how many had both of these components. You can see that really, it's an idea that's found its time. And you know this from the conversations you have and just being around at the moment, right? As his review was up to early 2020, and look at the number of comparisons that platform or MAMS trials that were starting up in 2020, and look how many of them are COVID-19. This is the approach that it really sort of found its feet in that setting. The red colours here are whether it's commercially led, grey is whether academic led, non-commercial. So you can see it's mostly still commercial trials using, and the yellow and white actually are whether they've got results yet or not. But so that feels a bit out of date. But it's finding its feet in other places. We've been involved with running or been involved in not just cancer trials, which is where we start off, but in some of those COVID-19 trials, in infections trials, which a unit also runs in TB and pediatric infections, but now in some neurodegenerative diseases, in motor neuron disease, multiple sclerosis, Parkinson's, in advising an abdominal disease, advising an eczema, the postpartum hemorrhage study with WHO is using this approach. There's lots of these approaches coming through, and we think there's lots of lessons. We want to make sure people do this right. Neuro, I just mentioned, he's also looking at the role of patient and public involvement and whether that's different. We'll come back to PPI in a moment. And the training needs for data monitoring committees, are they different? And particularly combining those two things. There'll be some findings about that to come out soon. So I'm going to talk about implementation. There's plenty out there about the statistical issues if you're using this approach. And actually Max and Babak Osku has been very involved in this work. They run a clinic now. If anybody's designing a trial, you want to talk about the statistical issues, you can book in with them. There's a very nice little video talking about it, and just sit down for an hour and run through. There's no fee, it's just a good conversation. And we run a short course, a half-day course about this in more detail. But I think it's more about the application to that real world that's going to support some of that uptake. And we tried our best to be sharing that information. We did one really about the implementation for the statistical issues, and Stampede came out of trials in 2009, and then about the practicalities and adding and stopping arms. Who did we give information to? How quickly did we get that to work within the oversight structure that we use? Who got information about the results? How do we handle that? For those patients who've been randomized very recently, could we re-randomize them, and in who? So we sort of went through a lot of detail. Max led on a paper a few years back now, looking at sort of 10 years of our experiences of running these sort of trials and some of the lessons coming through, short case studies. And then we had really focused on the trial management experience, the trial coordinators for running these studies, and for the data management services, for the people building the programs, thinking about the data. Voices that you don't hear in the literature enough, I don't think, but really about the conduct of this and the lessons we can learn. And now drawing together across some of the original, some of the sort of more mature, primarily oncology platforms in the UK. We've just had a paper that Sharon Love has led on that's literally come out in trials in the last few weeks. We've set out 50 recommendations based on 15 platform protocols at 8 CTUs, one of which is Precision Panc that Andrew Biankin is going to be talking about. He's connected into the paper. So I want to focus on sharing some of these lessons with you. And one of the key things to me, and this is really, in a way, it's true for everybody, isn't it? You know, the information that we give to people has to be right, but we have to tailor it. We have to find a way of tailoring that information and make sure it's fit. To me, information is like food. We all fill up at different rates. Having enough on our plate, what's too much for you might not be enough for me, or the other way around. And, you know, I think about this, you know, very much in the consultations I went to with my dad when he was unwell a couple of years back. He very much wanted to put his head in the sand and know absolutely nothing at all. Whereas me, I want to know absolutely everything. And the information that you want to give to make each of us feel happy to join a trial, I think would be quite different. Given if you're running a platform protocol, there's multiple questions, potentially multiple risks within there. You've really got to get that tailoring correct, haven't you? So in the Stampede protocol, we've done this with sort of modular, using modules, the multiple parts to the patient information sheet. We've ended up with a general patient information sheet, which we broke into parts 1 and 2. Part 1 almost setting out the rationale for the trial. Part 2 is almost the small print, the reasons why you would and the reasons why maybe you wouldn't join the trial. And then separate parts for the arms. And they could have those up front, but we were very clear to them, we know they may not read them. We'd encourage them to read them. But once they've been allocated to an arm, they've got to make sure that the information's there. But they can dip in, it's easier to compare and contrast across this, breaking it down in this way. And so trying to use a really good structure for showing that information as well. This formatting approach we've used is one set out by Peter Knapp in York, based on some research he did 5, 6, 7 years ago now. A really interesting work. This kind of narrow columns, big questions here. And what you'll also find at the end, I don't expect you to be able to see this, is a little table. The little table reminds you which other documents you need to go and cross-read to make sure you feel comfortable. And the front very much includes a summary, what's going on here, table of contents, and the contact details, which we think is really helpful. And we've had patients involved in helping us think about the design of the questions, prioritizing how we draw the information out, but also how we give the information back at the end. So here's Robin Millman on the left and David Matheson on the right. Two of the patient representatives here, they're talking about the results of the abiraterone study, really helping in a video think about how we give the information back to people. You'll notice this rather wonderful backdrop behind them of the City of London. That's not a green screen like I'm using right now. This is actually out of the window of what was MRC, Medical Research Council head office at the time. We'd had a fire alarm and our building was shut for a couple of days, and we went to do the filming there. They were up on the 13th floor with these wonderful views, and it was rather marvelous. And here's Robin again. Robin here is helping explain with some of the clinicians why we do multi-stage trials. And the patients are very clear. They love this design. Why wouldn't you do all your trials like this? Is their general refrain here. And here's Kevin Webber. Kevin was one of the patients on the trial. He's helping explain the results of the radiotherapy study as part of the interviews. It's really important to get that across. So let's dip in some of the lessons we have from Sharon's paper that we had come out recently as a practical guidance here. We've broken it down into these 15 areas. I'm going to touch upon a couple of lessons that have come out from some of these. I'm not going to go into all the detail for all of them, of course. Funding is really important. If you've got a trial that's going to be starting up with a view to adding something, then you've got to have something that's convincing enough to your original funder or funders that it does look reasonable that you're going to add something, that you know what you're going to do, that you've thought about it in a good way, and it doesn't look like a land grab. Is that a word that translates? It's not just putting down a flag and saying this area of cancer is mine now, all questions are going to do this. It's got to be, actually you've got to have a reasonable plan for what those questions are and how you're engaging the community, and a reasonable way of thinking about what that funding is going to be. So recognizing that funding is going to come from multiple sources, and that's not easy for a lot of funders to think about in some ways, because they each want a sense of ownership for what they're adding. So how do you do that? So you make sure that particularly when talking to companies, that yes, they're paying to support their comparison being the protocol, but it's not just their comparison that they're effectively funding or adding to the contribution to. Their money is going to the overall running of the trial. I guess like playing ground rent maybe on a flat, or a maintenance fee. Those are terrible words. I'm not sure the analogy quite works, but the principle here that it's being shared across. It's not just being spent on what they need to do. And actually, if industry needs you to do something extra in a trial, there's extra visits that are needed, extra monitoring that's needed for their assessment of their arm, then that's going to have implications for the control arm, and that's going to have implications for all of the other arms testing against the control arm, of course. So that becomes really important. The funding is in place to support this. And, we need to express to funders up front.
Matthew, something's going wrong with sharing your screen, I believe.
Ah, okay, let's try again. It did.
You were sharing a Teams meeting.
I was sharing a Teams meeting. Oh, that's
yeah.
weird.
You were sharing the Teams screen.
Ah, okay. Let's see. It looks like it's just jumped away from what it thought it was doing. Okay, hang on. Yeah, let me just turn the slides off and turn them on again. Let's see if that works. Is my camera still working? Is it part of that?
Yes.
Challenge? Okay. So I will stop sharing. Share again. How extraordinary. Okay. Are we back to, is that working again?
Yes.
Very good. If it happens again, let me know, because that's most bizarre, isn't it? Okay.
All right.
Excellent. Okay.
We see your slides again. Yes.
So when we think about adding an arm to a trial, you've really got to convince a funder actually why you want to do it that way as well. So we found it really useful to talk about it as the gains that they would be making. So you've still got to make sure you don't take on a question into a platform just because you can take on the question. It's got to be the question that's worth asking. So each time we try to cost up, how much would it be if we'd done it as a standalone trial? And then how much would it be if we did it within the platform? And you can do that for the original comparisons as well, actually, for you, the ones that you start off with, assuming you start off multiple arm. You can effectively be saying, by asking these questions together, we're going to save time, we're going to save quite a lot of money in this. It's often, I don't know, rule of thumb is something like, buy two, get one free, something along those lines. What about the protocol itself? I mean, clinical trial protocols can be complicated enough at the best of times, can't they? So, some of the trials that we looked at have used a singular protocol approach. Some have used modular protocol approaches, and you've got to choose the version that most suits you. I think if you've got a single point of randomization in your trial, like Stampede has, you probably want to be using a sort of a singular protocol. Whereas if you're using something that's a biomarker stratified questions, you're stratifying patients and there's different questions hanging down, then you probably want something quite modular. And I think that's what Andrew might talk to you about around Precision Panc. They've taken that kind of approach. And then you can think about the eligibility criteria for your patients too. What happens if somebody's not eligible for a particular comparison, particularly if you've got a single point of randomization? Can they join some of the arms? Could they be randomized between some but not all of the arms? And what are the implications of that? So think about the implications on recruitment if you're being strict, or recruitment if you're being generous. What will be the generalizability of findings if your populations are a little bit different in those comparisons? We're going to have to be very clear, but actually the Estimans framework probably helps us quite a lot in thinking about that. And what's the practical implementation issues at sites? Is it going to be easier to say, no, these people can't come in, or yes, these people can come in? So we've got to work through, and I think that's going to vary according to the questions at the time. We would also recommend that everybody in their protocol explicitly states that future comparisons may be included. We've added contracts just because contracts are always difficult, but making sure that contracts cover the longevity of a platform protocol, the fact that you might add that the time scale might drift on a bit is really important. And recognizing as well that the more collaborators you have in a protocol, then the more complicated the contracts can become. So you're worth budgeting a little bit for your legal team, of course. And safety, we have to assess safety in every trial. That's really important. But as you're thinking about what toxicities, what adverse events to score, then the more treatments you're testing, the more expected toxicities there are, of course, that you need to be doing for each of your assessing for each patient. And the burden of that can add up over time at sites, but also it requires very careful management of the reference safety information. Reference safety information was the key thing that the MHRA, the regulator in the UK, was testing everybody at during inspections in the last couple of years. So making sure that sites are on top of their reference safety information is really important. And then maintaining the control arm. If you've got a trial that lives for a long time, then there's a good chance that the what's the standard may change. In fact, you'd hope it would. So be prepared for that standard of care to change. I've gone back to showing you that plot of patients in Stampede. The blue lines here represent times at which we've changed the standard of care. So at first, all of these patients, the standard of care was hormone therapy with radiotherapy if you could. We encouraged it, but it wasn't mandated. And then we had data come out from one of our trials, an older trial, reported and showed that in non-metastatic patients, they should be having radiotherapy. So we started to mandate that as part of the standard of care. And then when the results of the docetaxel comparison came out, we said, right, have docetaxel. When the abiraterone results came out, we said, right, from here on in, you could have docetaxel or abiraterone. You tell us which, or maybe it's neither, and we'll stratify by that. So trying to take this into account. And of course, this does have implications for event rates going on. But this is the case for any trial where there might be new results. We need to be staying on top of this, but there's something about doing it within the platform framework that makes people think about it, which they wouldn't do for a traditional 2-arm trial. And maybe the longevity is the reason on that. And then I mentioned the trial management group and statistics here. We recommend having a dedicated comparison lead, a comparison chief investigator, maybe even a comparison co-chief investigator for every question that's going in. That can better support the chief investigator, and it really helps with the burden and the glory, I think is a phrase we'd try to use. And it's useful to have a senior statistician who's involved in the analyses and knows the trial well, but he's in the analyses, and one that's unaware of accumulating data. You're just planning from the outset to run that blinded, unblinded statistician model. Make sure your gatekeeping is good and well documented. Now, we know this can be difficult for ethics committees, and I know your ethics committee structure in Netherlands is a little bit different to what we have in the UK, but they're being increasingly exposed to these complex designs. We've found the Health Research Authority that looks after ethics committees in the UK to be a real ally for the implementation of new and efficient designs, but very few of their their committees have been exposed to platform protocols. So they invited us to go and run 60 to 90-minute workshops at 5 of their regional training meetings about adding a comparison. So we did this a while ago. So we gave a general presentation about the benefits and the challenges, and then we had structured discussion with them about what would they want to see if something was being added. If there's an amendment coming in, what do they want to know? So we asked questions about the planning of an amendment. What level of ethics review is required? Because at the moment, a brand new trial goes through a full committee, and an amendment often goes to a sub-panel, and the sub-panel might be the chair and like one of the lay members. So you could have like a chemist and a retired school teacher, and that's the only people reviewing an amendment. Maybe that's fine in some instances, but if you're adding a whole new trial effectively into a protocol, that feels inappropriate to us. So what should the level be, and what information do they need to know? Their forms didn't capture this, and what other aspects did they want? So we walked away with this with a sense that the initial level review should be very clear, and again, just like in the protocol, you say it might be a platform, you make it very clear to the ethics committee. And then for when you're making amendments, they need to know enough information for them to triage and have time to make that triage, whether it goes to the full committee or to a small subcommittee. So if the design, the aims haven't changed very much, the ethics committee might want to take the usual route. And if otherwise, it might be quite a full review that they want. But we also try to make it very clear, give them better information about what supports that triaging information. So, proposing covering letters for everybody with a table about what's changed, and does it result from, does it come about because of adding a new comparison, or is it from other amendments? Where are the changes, and can they work out what's a change that you need to make anyway? Because sometimes protocols need to change over time, and what's from adding. And then for recording, there are a table for them to record as well. Their information. So we've got a report about that online. MHRA have started, HRA has started to adopt this. It's in their standard operating procedures, and you can find a nice blog about it by Hugh Davis, who used to be the National Ethics Advisor in the UK. Let's just talk very briefly about non-concurrent controls. I just wanted to flag up, we had this data in Stampede where we showed a hazard ratio of 0.78, a survival advantage with adding docetaxel. We later showed a survival advantage for adding abiraterone with a hazard ratio of 0.63, which is even better, right? So everybody said, well, of course you'd use abiraterone given the chance. But we had a small group of patients who happened to have been randomized to either the docetaxel arm or the abiraterone arm, while both arms were open to recruitment. And I say small, it was 566 patients. That's far bigger than the original trials I worked on when I started out in trials 2.5 decades ago. So you could see the patients here. So what was going on? Well, actually, the population was a little bit different. More metastatic patients, like in the docetaxel comparison. The abiraterone comparison had slightly fewer because of that M1RT comparison, the one looking at just radiotherapy in metastatic patients. The population started to get a little bit older when the docetaxel comparison closed, because we had no age limit, but still people were not randomizing their older patients with the potential of chemotherapy. And PSA values seem to be changing over time too. And what we showed was a hazard ratio of 1.16, actually favouring docetaxel in these patients rather than abiraterone. Confidence intervals massively wide, but not pulling in the direction that people would have expected. And so, this story using concurrent controls makes me very much worry about anybody using non-concurrent controls as best. So, that's brief thoughts there. This is finding that, surprise the clinical community. So best we could do here, but the population might shift over time, whether you ask it to or not. The type of patient that goes into your criteria may change over time. And plus there's other changes, like what's available at relapse have changed quite a lot in this particular population. You want a longer example about that, there's a short sort of 10-minute talk. Very quick ad break from me before I stop. I mentioned the MAMS clinic. If you're interested in that at all, to talk to Max and Babak, they'll book an hour slot with you. Just reach out. There's more information on there. I would say we run a course on data monitoring committees, which is now online only a couple of times a year. We'll be doing it in February. If you know anybody that might find that interesting, new or starting out in sitting on data monitoring committees, particularly important for clinicians, I think, and highlight that the MRCCTU does now online only clinical trials, MSCs in clinical trials, and from this year, statistics in clinical trials. That's the end of my advert. Please forgive me for that. So look, clinical trials are the way we improve healthcare outcomes for patients. Including multiple questions in the same protocol has strong appeal And it's achievable. You can do this both statistically and practically. We know that those MAMS platform protocols set up a framework for additional research and future research. We have to do that in a sensible way. But there are these practical challenges to address, and experience-based guidance to set that out we think is really helpful. So we've just done this from a few trials. The aim is now to reach out to other people to share experience of those people, particularly the COVID researchers who have been banging their heads against walls in trying to do this over the last few years. And just to reiterate, the good patient and public involvement is really key in platform protocols. So thanks to everybody I've worked with on this, particularly these people named here. There's a QR code in the top right hand corner, which is a link to the list of presentation links to the papers and reports that we're mentioning here. And if you want more contact details about me or anything, my Linktree detail is listed here, or shortcut from there. Thanks for having me, Dara.
Then I would like to ask Peter, or Peter if I stay in English, to share his presentation and take us to the world of the statistical challenges in platform protocols.
I have to unmute myself. Yes.
Thank you.
Okay. Yes. Thanks for inviting me and giving a talk about this important topic. I will be discussing the more statistical challenges of master protocols, not only the platform trials and the multi-stage trials that Matthew talked about, but also different types of master protocols. Let me first introduce myself. I work as a biostatistician in clinical trials at the UMC Utrecht, well, my main interest is in innovative trial methodology, adaptive trial methods, and also multi-stage methods. Not only in oncology, but also in other areas, but predominantly in oncology currently. So the outline of this talk, I will first briefly give some introduction about master protocols and the different types of master protocols, and I will then, for each of the most important types, discuss the statistical issues, challenges, and well, the solutions that have been proposed and are there, and also some issues that remain open, I think, because I think there's still lots to improve and lots to discuss on these designs, which are fairly new. So what is, as Matthew already mentioned, a master protocol is basically just a single overall trial structure for running clinical trials, and the intent is then to evaluate either more than one investigational drug or treatment. It can be any treatment. In this presentation, I talk about drugs, but there are also master protocols for exercise interventions and other type of interventions. So it's not just restricted to drug research. Also master protocols can intend to evaluate interventions in more than one type of disease or one type of cancer, for instance. The advantage of using a single infrastructure is mainly the efficiency, the logistics. Well, you have an infrastructure. It's easier to incorporate new trial arms. For instance, you have a protocol which is standardized for all treatments, so there are many advantages. Well, the purpose can be quite different. You have master protocols for early phase research, but also for more late phase research.
Something's going wrong here again.
Yes. Okay. So so first start with
we still can't see your slides.
Okay. Then I.
Yes.
Yes. Okay.
Yes. Thank you.
Not sure what went wrong, but well, two important types of master protocols are the basket trials and the umbrella trials, and they look well, when you see the diagram, they look similar, but they basically quite different, and also pose quite different challenges in terms of the statistics and the methods used. So basket trials, they evaluate a single targeted therapy, but in, for instance, in multiple diseases or multiple cancer types. And because the therapy targets a common genetic mutation, which might be common in the cancer types, these patients that have either a lung cancer, colorectal cancer, or other types of cancers are combined in one basket trial. Each receive the same targeted therapy that targets the mutation that is present, and with the idea that these patients respond similar to the drug, or at least all benefit from this targeted therapy, nonetheless, despite that they have different, the cancer in different locations. In the umbrella trial, they typically restrict to patients that have the same cancer type but have different genetic mutations, and then for each genetic mutation, they use a specific targeted test drug. So umbrella trials typically involve several test drugs that are evaluated each in a single genetic mutation, whereas a basket trial typically evaluates only a single targeted therapy. And in the diagram, I did not draw a control arm or a control treatment, but there are basket and umbrella trials that also involve randomization, also include a control treatment, a standard of care treatment for each of the cancer types or for each of the mutations. A more elaborate form of a master protocol is a matrix trial, where basically the elements of an umbrella trial and a basket trial are combined. So they typically combine different cancer types, but also different mutations, and basically consider a large population of cancer patients. And some drugs are evaluated in multiple subgroups, and others are only evaluated in a single subgroup. But I will not specifically discuss the matrix trials here. Matthew already mentioned and talked about the platform trials, the multi-stage, multi-arm trials, where interventions can be added throughout the study and also stopped for futility, where typically these are characterized by many interim analysis and the possibility to basically facilitate continuing research and adding interventions and looking at new interventions for the same disease over a long period of time. So, about the master protocols are used and are becoming more and more common. This is quite not the most recent overview, but there's a landscape analysis from a few years ago. There, the majority of trials with the master protocol were the basket trials. These were mainly exploratory and in phase 1 and phase 2 research, and considered non-randomized studies, so without a comparator arm. Umbrella trials and platform trials were slightly less common. Umbrella trials were also mainly exploratory as the basket trials, but they did more often involve a control arm and randomization. And I think the number of platform trials at that time was still quite small, but increased quite a lot during the COVID pandemic. But typically the platform trials were randomized studies and involved multiple arms, and indeed were mainly used in a confirmatory phase 3 setting. So I think the less statistical challenges are there for the umbrella trial, so I'll start with that. Because the nice thing about the umbrella trial is that you evaluate different drugs in different mutations, and you want to make only an efficacy claim, one efficacy claim for each test drug. So you test the efficacy of drug 1 in mutation 1, and that's it with regard to the efficacy of drug 1. So an issue like multiple testing, which you typically have when you do multiple hypothesis testing in one single trial, is not it's not that present here. Here I think it's acceptable to not control the family-wise type 1 error over the whole umbrella trial, but you could stick to controlling it within the for the specific test drug. So, of course, if you do interim analysis for the test drug, then you need to make sure that your type 1 error is controlled for that test drug, but you do not need to control the type 1 error over the whole umbrella trial. A disadvantage here is that, and it's also something with the basket trials, is that the subgroups may be small, especially if you have rarer mutations. The numbers can be quite small, and then you have limited power to detect an efficacy of the drug. So that, I think, is the most important limitation of an umbrella trial. So the basket trials, they are generally more challenging in terms of analysis. They typically lean on the assumption that basically it's the mutation that is predictive of the treatment effect, and it's more relevant, basically more important than the cancer type itself. But often we do see heterogeneity in response rates between the cancer types. So the overall response rates are typically not the same in the different cancer types, and there might be cancer types that have good response rates and cancer types that have slightly worse response rates, even though the treatment is targeting the same mutation that is present in all of the cancer types. So here the debate is often whether we should do a pooled analysis and look at a tumor agnostic evaluation, so ignoring the cancer type and making a statement about the targeted therapy across all cancer types, or performing separate tests for efficacy in each of the baskets. Now, if we do the latter, then of course we do multiple tests regarding the same test drug in the same trial, and then we need to account for the multiple testing and need to do a correction. And several approaches have been suggested for dealing with basket trials. The old and naive approach was to just do a pooled analysis, so tumor agnostic analysis, assuming that the overall response rates were the same across all baskets, so they did not differ between the cancer types, and you calculate just one effect estimate and you assume a full homogeneity. Well, the advantage there is that you can pool all the data, you can use all the patients, but of course, if there's heterogeneity, it's the pooled estimate may not be that informative. The other option in the frequentist setting would be to, as I should suggest, to only do basket-specific effect estimates, but then you need to account for multiple testing, and also there you might have the problem that these baskets contain very few patients, so then you have an issue with the power of the study. So to solve these issues, several Bayesian approaches have been proposed. The first one was using a single hierarchical model that basically assumed that the overall response rates could differ between the baskets, so it could differ between the cancer types, but there was one single distribution that covered these response rates, so it did take into account the heterogeneity. But in the latter years, these methods have been criticized a bit, and there's been the focus has shifted towards so-called dynamic information sharing methods in a Bayesian setting, and these, what these methods basically do is they try to identify subgroups of patients, subsets of baskets that have similar response rates and group them together. So they do account for the heterogeneity, and they also have the advantage that you could, for instance, in the figure that you see on the right bottom, that would be a setting where we have like 3 different subgroups of baskets. Subgroup 2 is a set of baskets, a set of cancer types in which there's quite a high overall response rates, whereas one, basically you do not see responses in those cancer types. So, as I said, a single pooled analysis in a frequentist setting, then typically the power is high, but you do not have specific statements regarding each of the cancer types, so you only have one statement, and you do not take into account the heterogeneity that there might be. The basket-specific analysis, then, typically you have low power, so it might be difficult to show efficacy. And I think the Bayesian exchangeability methods or the dynamic borrowing methods also they do have advantages. I think because they enable identification of the subgroups of the sets of baskets in which the treatment has a response. It also allows quantification of the amount of evidence that there is for tumor agnostic efficacy. So if you only find one subgroup that has equal response rate, then it's quite strong evidence for tumor agnostic efficacy, so that the treatment works irrespective of the cancer type. But the only disadvantage that I see is that it's quite a data-driven method. You just pull baskets that have similar observed rates only based on the data that you observed during the trial. So at least I think this needs to be supported by clinical knowledge or expertise. So really, or at least or separate confirmatory study afterwards. But that's the only disadvantage that I see with these Bayesian methods. Many basket trials and many umbrella trials, as I said, were designed to be exploratory studies and only are single arm, do not involve a comparator. But sometimes they are used for for asking approval for marketing authorization or conditional marketing authorization. And the problem there then is that you do not have a comparator. Then typically you look at the overall response rate and the duration of the response, but which might be good or above the level that you want to see. But to have an assessment for the longer term efficacy is quite difficult for these in these settings, especially when you look at overall survival or the time to event endpoints. Because there can be something that we call lead time bias, and that occurs when the standard of care changes over time. For instance, and that specifically holds for studies that have a longer period of accrual, and or when you compare to studies that historical controls that are not the most recent. So then it can be that for instance, at the time of detection of the cancer is shifted between the historical cohort and your current sample, then you might overestimate the overall survival in your current study just because your patients are diagnosed earlier, or because first or earlier line treatment has ended earlier and patients are included earlier, started treatment earlier than they did previously. So comparing time to event outcomes to historical cohorts is very, very difficult. And that's also quite a challenge when you consider these basket studies, which are typically single arm, and when you want to say something about the longer term efficacy outcomes. And there's also still the issue if, even if there's homogeneity of your response rates across the baskets, then that does not automatically mean that there will be heterogeneity across the across tumor types for the other outcomes. Still, there can be other factors that are more important for overall survival than the duration of response and the overall response. So I would like to stress the need for a comparator also in these basket trials, even in earlier phase studies. And there's also a nice recommendation paper from the French HTA authority, which gives some suggestions for or recommendations for basket trials. So to always include a randomization to a control arm if that's possible, to not only consider the response rates in combination with the duration of response, but also other clinical relevant endpoints, such as quality of life and overall survival, progression-free survival, correction of multiple testing, as I said. Even if you do a pooled analysis, then of course you should always show the heterogeneity across subgroups, for instance, using a forest plot. And if you only if you do not have many results about overall survival, then it's important that you establish or show that there is at least a correlation between the duration of response and the overall survival. And something that, well, that's not directly related to the efficacy of the drug or the treatment is also the quality of the assay that is used for the patient selection, because typically basket trials, you identify patients for the treatment using a biomarker, and that has to have good sensitivity and specificity, for instance, to select the right patients that will benefit from the treatment. So I think I'll briefly go over the platform trials, because Matthew already discussed them. Typically, the issue there is that you do multiple testing interim analysis. That you can do that by either using Bayesian methods or frequentist methods. Some of these multi-arm, multi-stage trials also use adaptive randomization to allocate more patients to more promising treatments. That is, of course, more ethical, but it also poses some challenges for the analysis and for checking whether the family-wise error rate is controlled. Well, I think the most important issue here is indeed the discussion whether we should use only concurrent controls or also the non-concurrent controls in the analysis. For instance, in the figure here, you see that for intervention 4 starts quite late. We do have patients on standard of care in the first 3 intervals of the trial. Of course, this brings a lot of information, but we could also restrict to only using the patients on standard of care in the same period as the inclusion period for intervention 4 for the first interim analysis. The use of the non-concurrent controls that potentially introduces bias, especially when there are temporal drifts in the outcome or in the inclusion criteria, or if the patient characteristics change over time. So, and of course, there's a trade-off, because if you only restrict to the concurrent controls, then you have a lower risk of bias, but you also have less power. And recently, there have been some methods proposed for incorporating data of the non-concurrent controls. What they typically do is they use some modeling, either Bayesian or frequentist or network meta-analysis models to model the response over time, and they basically adjust for that. You could also use a more simple analysis where you adjust for the patient characteristics that are changing over time. But still, the issue is whether your type 1 error is controlled or not. That strongly depends on whether the model assumptions hold. And so the best option here is to do the primary analysis only using the concurrent controls. You could do a analysis using non-concurrent control data, but and adjust it for temporal trends, but only as a supportive analysis. And when you select the method to adjust for the time trends, then it's ideally take into account the population, the disease, and the endpoint and clinical expertise to decide on what's the best way to adjust for the time trends. Is it a step function, or is it another type of a model? Adaptive randomization is used quite often, especially when there are multiple arms. Of course, that helps you quickly identify promising treatments arms and get a lot of information on these treatments. A side effect is that you only collect very limited data on the treatment arms that perform bad at the beginning, so you cannot make firm statements about them. And because you test multiple treatments in a single trial and compare them against the same comparator, as multiple testing correction is needed, but mainly because you use the same control arm for all comparators. So to conclude, I think most of the challenges are there for the basket trials and the platform trials, mainly the heterogeneity in the basket trials and the time trends in the platform trials. But there still are some open issues with regard to the long-term outcomes. Of course, you can solve these partly by including a control arm in the basket trials and in the umbrella trials whenever feasible, or show that there's an association between the duration of the response and the other clinical outcomes, such as overall survival. They are focused mainly on efficacy, but of course there's also challenges for safety evaluation, especially if you have small subgroups in the basket and umbrella trials. Then it's hard to find any safety signals in these early phase or in these studies, and that also is quite a challenge to say something about safety based on these studies, because they're typically designed as early phase study, but sometimes submitted for approval for marketing authorization, and then there's limited information. So, and these were basically the references that I used.
All right, thank you. If there are no further questions, then I would like to invite Andrew to start off his presentation. He's going to discuss platform protocols from a clinical perspective with his experience in pancreatic cancer, if I'm correct.
Correct. Can you guys see my screen?
Yes, we can see your screen. Thank you for sharing. And well, the floor is yours.
Okay, thanks very much for the invitation to share some of our challenges, successes, our pains, our woes, and all the things that come with generating a platform. So if people wanted to read more detail, we published this about 7 years ago or so, and it's sort of by way of background, but I think most people understand that background there and some of the challenges in designing these sorts of studies and trials and platforms, etc. So one thing I will say is that the statistical design of your actual trial itself, or the trials that you run, are probably the least problematic. We now see that there are so many different subtypes of cancers, and you're always going to be underpowered in many of these subtypes and segments. What is the solution to that problem? Do you adjust your study? Do you open 3,000 sites to recruit 30 patients? How do you do that? Randomized is always better, but sometimes randomization is not possible. For example, we wanted to target the KRAS wild type segment in pancreatic cancer, which is about 8%. And a reasonable randomized study is about 35 patients in total. Not a lot of patients, but it's about the bar of your signal that you're looking for. And there, of course, there's orphan indications and other approaches there for smaller segments and subgroups. That often depends on the owner of the asset who's running the trial, whether they do have orphan designation or not, and what sort of trial that they need to run. So just to think back, people often have this debate about whether precision medicine or precision oncology is actually benefiting, and maybe we should go back to boiling sponges from the bottom of the ocean and bark from trees and develop drugs in that particular way. But this is just to remind people that the first example of precision oncology targeting biology was first reported in 1896. I am a little bit biased because it was reported from Glasgow and it was a surgeon, George Beatson, and he removed the ovaries of a woman with breast cancer to suppress the growth of the tumors. And people often think the beginning of precision oncology started with Herceptin, but the most successful therapy for cancer that, say, helped and benefited more cancer patients than probably all other therapeutics combined was tamoxifen and its derivatives, which is a targeted therapy developed in the 1970s. And what you can see there is one experiment that someone did, and that was use tamoxifen to suppress the perineal flush of a baboon in heat, demonstrating its anti-estrogen actions. So in principle, targeting biology works. The challenge is for estrogen receptor positivity in breast cancer, it's about 70%. Even if you do an unselected study, you're likely to see a signal. Most of these segments in the biological complexity is breaking down these segments to much smaller sizes, and hence the challenge, and hence the benefit of platform trials where you can find opportunities for all patients by having a pharmacopoeia of potential trials and therapeutics that start to come in piece in, and so you can offer patients clinical trials at the right opportunity, already prepped for these trials, rather than waiting to the after effect. And we see this problem particularly in pancreatic cancer. We have many drugs. Most of them don't work very well. They work quite well in a small proportion of patients, but we can't predict them ahead of time. So therefore, we keep accumulating these drugs, some that are often not used, and the overall effect size is very, very small. We have some that target small segments like olaparib and pembrolizumab, but in general, the all-comer studies have a very poor signal. Of course, we can see these in pancreatic cancer, and you know all of this, but if you did a study for targeting BRAF mutants in pancreatic cancer and recruited 50 patients, you'd be lucky to get one patient that was a BRAF mutant if you didn't test for them ahead of time. And of course, the challenge with this, by the time you realize this, that the indications moved on, the patent life is too short, and so chasing a 1 or 2 or 3% segment in pancreatic cancer just doesn't have the commercial viability to develop it in that way. Here's an example of a pancreatic cancer patient which we retrospectively identified. This is from years ago. This is from 2010 or 2011. We were doing the International Cancer Genome Consortium project then. And this is a patient that was 2 months away from death. This is the peak of their CA19-9. They went on to platinum therapy just, for no specific reason, just through the choice of the clinician. Their disease virtually disappeared. They continued to live. They had an unusual recurrence pattern with cerebral mets. They went on to platinism again and a PARP inhibitor, which again had a response. And we identified in retrospect that they had a HRD signature in their genome. And so when you do get it right, even in bad diseases like pancreatic cancer, this is somebody expected to survive 2 months, lived for 5 or 6 years, more than they were expected to, which I would call a pretty good result in pancreatic cancer. So one of the things we often say in this slide, I put this slide first up about 10 years ago, maybe 8. We'll be wondering where all the big ideas are. Some people will wonder if science is slowing down and we've eaten all the low-hanging fruit. And I would say that this is correct, because we still build only the engines that allow us to answer very, very simple questions. If we don't build the new engines, platform trials are one of them, then we're not going to discover things that are new. Just doing more and more of the same thing on bigger and bigger numbers has its benefits. But if you need 10,000 patients to prove that a cancer drug works in your clinical trial, then the effect size is so small that it probably won't be used or approved for therapy based on the qualities. So, as we all want to know, we want to move from single to multiple gene tests. We want to be able to identify our patients, test them, and then put them onto studies. Now, if you are thinking of only running one of these trials, think of the disappointment for the patients, think for the disappointment for the clinician, think of the fact that the tissue has disappeared. You need to rebiopsy, and for diseases that are rapidly progress, basically patients die. So we built this system where we needed to build an engine, which was the Glasgow Precision Oncology Laboratory, which allowed us to do the testing that we needed to do. We built a platform called Precision Panc for pancreatic cancer specifically, and also another study which I haven't gone into, which is called Imagine for early phase clinical studies. And then it allowed us to accumulate data and knowledge with the new definition of precision oncology. It's not just finding a drug for the patient, it is about understanding the molecular pathology of the disease, so that when you have another opportunity for a therapeutic, you've got a smoother path to introduce that in a trial. And then, of course, aggregating those data in an international platform called ARGO from the International Cancer Genome Consortium, and sharing those data and identifying where those segment sizes are, where you can start to translate. So what we needed to do was be able to have this system in place as a broad-based system, but specifically today we're talking about the trial itself. So the first thing we needed to do was to build a test, because the tests that existed were too expensive, out of date, and we just weren't funded to run those tests. We couldn't pay £3,000 for a foundation medicine test, for example, per patient. So we built our own test. We didn't want to, but we had to at the time, and we dropped the prices down to about £300 a test. And we drove down the tissue requirement to being 50 nanograms of input DNA. We could probably push down to 10 nanograms if we had to. And the very small, ratty little biopsies that were formalin fixed were still sufficient for us to get a decent result. And we get essentially, once you get DNA out, you have a 98% success of having the test result. And then we return a test result to every patient that participates and their treating clinician. And if there's a trial on the platform, we recommend them for the trial. If not, we find other trials and we try and build partnerships. This is the other challenge we had with our funding bodies. They would say, oh, well, you don't have a trial for this. Why are you testing for this? And we'd say, well, there's an industry-sponsored study, in the hospital around the corner. And so if they don't fit for us, then that's a good thing for the patient. They didn't seem to understand that for some reason. And that was one of the other challenges. So when we built the platform, we had to think about all of these factors, single gene tests, multi-gene tests, not enough options for patients. How do we move from these single trials to develop a platform that's going to be useful? And then how can we start to instill that in the system with expanded access programs, etc., to start to garner some real world data where we didn't have the degree of investment to analyze these patients in great detail. And one of the underwriting concepts from our patients, and this is important, speaking to the patients about what they actually want, is that they want an option. For a disease like pancreatic cancer, they want some novel option, some novel clinical trial. And the more options they have, the better. And so very critical for that is that we had to think about how we designed this platform to make sure we could really address these patients' needs. And so the core principles of Precision Panc were to how do we use current treatments better? In pancreatic cancer, FOLFIRINOX, probably the pinnacle for the athletes. Only 25% of patients receive FOLFIRINOX, and of those, probably only about 10 to 15% get a meaningful result. So we also know that pancreatic cancer can change with treatment. Many cancers will evolve in treatment, and we have little knowledge and understanding of what happens to patients that get into clinical trials. How do they compare to the usual cohorts? What happens to them after they leave their first-line study, particularly if they have a response? What does the phenotype look like? And also, how are they going to respond? And this is more important for other cancers, is that, what's a lethal cancer versus a non-lethal cancer, prostate cancer, for example. And then how can we use what we've learned to accelerate therapeutic development with closer involvement with the clinical setting without having to constantly build overriding infrastructure and pay for it? So we had to build the system, and the principle we were approached was a master protocol. So a master protocol gave us the opportunity. It's like a funnel. Bring the patients in. There's challenges with that because you can't just bring patients in unless you have some options. And this is one of the challenges of the observational study, in that we do run an observational study in that once a patient is in the platform, if for some reason they're no longer eligible for these studies, we continue to follow them. And we observe them because people, as you know, people can be excluded for all sorts of different reasons. We have their molecular subtype and we can come back to them. The patient might choose, they might not want to go onto the trial that's recommended for them, but they have a subtype where they might be eligible for a second-line study that we have open. And so we keep tracking them, and we're getting some informative data just from this observation because they're already in. You've expended the resource, you've expended the energy. You've asked the consent to be able to follow the patient and can communicate back to them if you have a study that has opened or if you come across a study that they might be eligible for. Working through this is a very local scenario. We had to have two consents in place. One was the pre-biopsy consent and one was the post-biopsy consent, because of course many patients that get biopsied at this early stage, they don't have pancreatic cancer, they might have a biliary cancer and ampullary cancer. And this is probably one of our biggest wins, is that we started having the conversations with the patients early, at the point of first interaction. That way you can prepare the patient for your trials, and the turnaround time question for the molecular testing disappears, because we can get a molecular test done faster than most hospitals can get a CT scan done in an elective setting, because you've taken your biopsy week 1 and you've got another 3 or 4 weeks to deliver the test result. So this is probably the overarching slide that tells you how we've developed this. So when you think about this in the first line setting, you want to have enough open indications so that most patients have an option. So the master protocol encompasses pre-first line, also comes in pre-second line, and that should be plus or minus biopsy. You don't always need a biopsy. Just being very pragmatic. We know that tumors might evolve, but the context is get biopsies where you can. So the one thing we needed to do is have a catch-all study, which says we have an offer here for a trial in this indication where most patients will be eligible, because otherwise you're going to screen through them and most of them won't get a trial because of your molecular segment sizes. We were heavily criticized for that because they said it's not a precision medicine study. But if you don't do that, you're going to get very low uptake. There needs to be a study where most patients can participate. It needs to be relatively novel. This study is nearing the end of recruitment. We're a bit slower than we'd like because of COVID, but it's essentially comparing Folfox-A versus Gemabraxane. Some people might think that's not terribly exciting, but it is, as the opportunities there is that you can standardize your first line treatment. You understand the population of patients that are coming into the study, and you understand the populations of patients coming out of the study, and you also give them the option, because they've had a molecular test, they'll be ready for second line studies as well. We're looking at external partner trials where if a patient isn't eligible for one of our studies, we try and find them something through some sort of relationship. And of course, even if they elect not to go on to study, they go on to standard of care, and we still track them because there may be an opportunity for them in second line. We have a study 005 for locally advanced disease, a different indication. And we also have a neoadjuvant study, which we did close recently because the question had changed. It was not a therapeutic question, it was a tumor evolution question. We're having negotiations as to how to introduce that study. And of course, because they're first line standard of care treatment doesn't mean they shouldn't be able to enter the platform. And they're the mechanistic challenges, because if you're first line, the NHS will pay for the biopsy. If the biopsy is insufficient, you have to pay for the biopsy yourself through your research funds. And if they come from an external first line standard of care, unless you've got a partner that will pay for the biopsy, you also have the money to pay for that. And then after that, what we just put in was a second line umbrella, which is very much looking at targeted studies. The biggest challenge we've had there is that you make a deal with a pharma partner. By the time you go through contracting through the university and the hospital system, something might change and the priorities of the pharma company may change. The drug may be sold on to somebody else, in which case you have to start the discussions again. So the most challenging thing about this is not the statistical design of any of these studies. It's just navigating the world in order to make this happen. One of the successes we did have in the second line is that we made it one study and we just insert cassettes or amendments, which decreases the turnaround time from 18 months or 2 years to about 3 months or 4 months. So we can go from zero to making agreement and then starting a study within a matter of months. But unfortunately, it takes a long time for the contractual stuff to get done. And we're working hard with our pharma partners to make sure that that happens. The other challenge is getting 2 big pharma companies in the room is difficult because they often match each other's assets. And so that poses challenges. So we were fortunate enough to have what was then Celgene and BMS and AstraZeneca involved because they had non-overlapping assets. But then the more success is working with biotech companies like RedX and others. I'm not saying that this is easy. There's challenges that are not scientific every step of the way. And so if you're thinking about this in a different way, you have a funnel that captures your patients. You have to have something on offer for the patients, otherwise it's unreasonable to recruit them into the master protocol. Plus, you'll probably burn so much money on doing your molecular tests and not that many people will get into studies. And then you map out what the indication space looks like. And then you understand how you can position certain therapeutics, and you go and make deals with your biotech or pharma partners. The sort of studies we've landed on for the signal-seeking studies, I won't go through this in detail. Essentially, this is originally a Bayesian design where the magic number was 21. Single arm looking for objective response rates. And that's where the game was in pancreatic cancer, and that's where we focused for the novel liver. But every indication will be different. But typically, if you have 7 patients and they haven't responded at all, recruiting another 7 isn't going to help you. Unlikely to, statistically speaking. But we ran a study like this called RUCAPANC. And in the first 15 patients of the 21 patients we were meant to recruit, there were no responses. So we stopped the study, but another 4 patients had already started their treatment with rucaparib. And all of those 4 patients had, good, robust PRs. So even statistical Bayesian thinking isn't foolproof when it comes to probability. So when you start to get throughput into your platform, then you can start to understand the feasibility of doing certain studies. So this is a bit of work that we did looking at what is the prevalence of DNA damage response deficiency and what's the predicted prevalence as patients go into second-line therapy, whether they respond or not respond. There was an anticipation of a platinum response prior to putting them on a PARP inhibitor, similar to the POLO study. And so we had to map out those numbers and the feasibility of how these numbers are going to fall out before you start your study to understand whether you need to open more sites, whether it's not feasible at all, whether an orphan indication is better, maybe changing your parameters based on the data you have. So a really deep dive on understanding the pathways that either you have data on that you can predict, or you infer from that and take an educated prediction. So this is something I'll upscale a little bit further. This is not just for platforms. This is work I did for various pharma companies like Genentech and Pfizer, etc. And this is mapping out practical prevalence. So if you don't have a platform where you're running studies, say this, I did this in the US, but you can map that out across Europe. What are the numbers? So on the left, you'll see what are the clinical inclusion/exclusion criteria, then map on top of that the molecular criteria that exist. Then you look in your population where these cancers are distributed. In the top right, you look at, well, what is the current patient flows? Where's your competition positioned itself, if there is any competition? And then you can look in the bottom, in the middle, you look at how many trials are currently open competing for the same patients as your trial. And the size of the box is the expected recruitment numbers. And it's really interesting, if you look at these trials that are running, it's almost like a mutual exclusivity map of sites. So because sites won't open competing studies. And so these are all not so important in a platform study that's run academically, but critically important if you're developing a drug in the commercial sector. And here's a summary of how you work through this particular analysis. I haven't done it in Europe because nobody's asked me to do that, but I've only done it in the United States. And then the other aspect is this sort of real-world therapeutic testing, expanded access programs. They're becoming far more to the fore now. They're even listed on clinicaltrials.gov. So if you look at those boxes, each one of those represent certain populations. One proportion is within the inclusion criteria and the rest are outside. So how can you offer those patients something yet glean sufficient information by de-risking your study? So there are examples of trials that have run expanded access programs. That means it doesn't pollute the trial itself. The trial data you can still file. If the expanded access program is saying extends into somebody with, say, borderline renal function or over a certain age or a slightly poorer performance status, if the trial is positive in that population, you can co-file with your trial population and expand your indication. That hasn't taken off as well as it should for a number of different reasons, but certainly some companies have capitalized on that to do that. And we should do that in the academic and the public sector as well. Where are we at the moment? This is the map of patients that have entered the study. There's about it's actually over 600 now. There's a bit out of date. Then we register these patients when we've got a sufficient biopsy and the biopsy, and we're able to have a meaningful result. And this is where the performance status attrition in pancreatic cancer happens. So we've gone from 429 that are potentially eligible to 250. So you get about a 50% attrition. We try and understand why that's the case. And it's probably mostly performance status, but as you can see, there's a whole bunch of different reasons there that one can address as you build your platform and you evolve it moving further forward. And the information we're getting, the data we're getting from this is wonderful. We can see that the genome and the transcriptome in metastatic disease, this is metastatic disease as compared to primary disease. There's not a lot of difference. We anticipated there would be differences, but there's not a lot of difference in the major ones. There are certainly some small changes in the minor ones. For example, RNF43 is suddenly up to 8 or 9% in the metastatic disease, but it's only 4% or 3% in the early disease. And we can recapitulate these subtypes. Remember, these are very small biopsies, routine formalin fixed paraffin emitter biopsies, and this is a new platform called TempoSeq by Bioclavis, where we're able to recapitulate the molecular subtypes we see, and we're trying to work with them to develop a diagnostic because there are clear differences of therapeutic response that are at least hypothesized in the preclinical section. And so you can build a lot of basic and translational science on the back of this platform. And I'll just finish with, how do you share data? So because we run the International Cancer Genome Consortium, we're sharing our data through the ICGC. We welcome platforms to become members of ICGC, contribute their data and benefit from the fact that they don't have to worry about how they have to share their data because all of the hard work's been done. And it's all done in a GDPR acceptable way that's acknowledged by most funding bodies and most notarized bodies and organizations for sharing your data. And that's just our website if you'd like to take a look at it. And we've evolved into something very patient focused, and these are the countries involved so far. I'm missing a couple because this slide's a little bit out of date. And with that, there's a lot of people that contribute to this. There's about 250 Precision Pank members around the UK. These are the key people in the leadership. And of course, the various funding bodies did need a lot of money to make it happen. And thank you very much for listening.